This is a practice talk on the dynamics of eating rates and obesity. I want to begin my talk with my background, which is my training is in the mathematical sciences, and I currently teach mathematics courses at both the undergraduate and graduate level at Montclair State University. However, all my research is conducted in collaboration with psychologists, medical doctors, and professionals in the field of obesity to look for solutions to treat, reduce, and prevent obesity. So I hope that not just content will be delivered today, but also a philosophy of how we integrate the different disciplines to come up with really novel solutions and advance the field. So I was introduced to eating rates in a very odd way. When I first came into the field of obesity, Corby Martin, who is a psychologist at the Pennington Biomedical Research Center, introduced me to eating rate studies and gave me a paper of his, which I'm going to show you today, on eating rates. And he said, he made a comment at the time that, you know, I wish we really knew you back when we wrote this paper because we could have used you to really understand these parabolas. And I thought to myself, it was a little like hurt. You know, I work in much more advanced topics than parabolas. But it turns out that this area is really interesting. So I want to show you the biology and what is known about eating rates largely from animal studies that were conducted in the early 1900s. And then I want to move to that to what we call first principles modeling. First principles modeling differs from what you might be familiar with, which is statistical modeling or curve fitting. And so I want to give a pitch for what first principle modeling gives us and what a good model is, and how we advance the field of modeling. Once I've developed a model that describes eating rates, I want to show you the cool things we can derive from that model and and the applications we have from them. One of the biggest applications is that when someone eats, we know that they have a fast phase and a slow phase. And there's actually a point that switches between the fast phase and slow phase. And so I want to show you how we can actually use the model that I've developed to calculate that switching point between the fast and slow phase and what it tells us about the eater. So let's begin with a very interesting history of eating rates. First, I'm going to take you to the modern day. We know that treating obesity, one of the ways that is prescribed is to eat mindfully. This is a book by Susan Albers. There's a great wealth of public feeling towards eating mindfully. And there's a perception, I think, that people with obesity are not eating mindfully. They're eating fast and not paying attention to their food. And all they have to do is eat slowly and things will be corrected. While this is great, a great concept, and hypothetically it makes sense, I'm going to actually show you there's no evidence, very little evidence, that there's something behind all of this scientifically. And it's not for lack of thinking about it. People have been thinking about this for a long time. Why? Well, how much we eat is really important because if we exceed the amount we eat with the amount we expend, then that's when obesity occurs. So getting a handle on how much we eat is important. Well, this depends on several things. We can break it down. How fast we take a bite or our eating rate is important. It contributes to how much we eat. The mass of each bite, so some people may take small bites and some people may take big bites. So the mass of each bite is a variable that contributes to how much we eat. The energy density of each bite, so eating spinach quickly is different than eating a donut quickly. And how long we eat. Those are the variables, if we want to break it down mathematically, that contribute to how much we eat. So understanding how quickly we eat or how quickly we take, how much mass is consumed per minute, those kind of variables are important in this bigger picture of how much we eat in totality. Early investigators were interested in some particular questions that involve eating rates. And that first question was, what drives someone to eat in the first place? Obviously, we can I asked my kids this question and they said, well, you're hungry. Well, what makes you hungry? What is it that signals to you that you are hungry and that you should eat? And the second is, obviously something stops you from eating too, because otherwise your stomach would burst. So what drives you to stop eating? These were the initial original questions that led investigators to try to understand the drive to eat and the drive to stop eating and eating rates in general. And finally, the ultimate question is, if I know the answers to those two questions, can I change someone's eating behavior? In the early 1900s, when they're first exploring these ideas, there was not really a epidemic of obesity around. So they were probably asking these questions more from a basic science viewpoint. But today we are looking at these questions from a different viewpoint. We want to know if there's ways that we can treat people when they have been affected by obesity. This is one of the early rat studies that were published by Skinner in 1932. Look at the title of his paper, Drive and Reflex. And here he's plotted the amount of pellets that have been consumed by a rat over time. I think this is time in hours. I don't see the units on this axis. But in this scenario, Skinner had rats inside a cage, and they had to obtain a pellet by pushing on a lever. So they could actually record the number of pellets that were consumed. This is actually harder to do than it appears. I have a faculty member at Montclair State that works with mice and animals, and he told me that there's a lot of waste, and recording that waste is really hard. I guess today we have cameras, so we can actually see if the pellets fall to the floor and are consumed later, but it's not easy to actually record. I do want to point out that in this caption, you might be able to see that he has here n is equal to kt to the n. He's trying to actually model this curve with a function, and it's a curve fitting exercise. And so the math is linked right up to the actual original analysis. I want to show you some more figures, but before I show you those figures, I want to point out that so much was done pre-1970s with animals that really contributed to our understanding of what drives individuals to eat and what drives individuals to stop eating. And I'm just showing you highlights of some interesting studies, but to really delve in the literature, it's quite fascinating. This one was a picture that I hope stays in your mind during the rest of my talk. These are rats that were deprived of water. And you can record the water intake by looking at they suck water out from these devices, from these little containers. Again, there's going to be waste. These are not easy things to do, but you could record the cc's of water consumed by also recording the waste, what was thrown out. And these particular rats were deprived of water for different amounts of time. And what you can see is that the general shape of the curves, of the water intake, are the same. However, the more that the rats have been deprived, the curve starts to look very different. So for example, here, this is the fast phase I'm talking about. The amount of water that's consumed quickly takes longer. So the fast phase is longer for those that are more deprived of water. And moreover, this is a number we call Emax, the maximal possible intake for that particular situation. And we see that rats that have been deprived level off around 30, 33 cc's, whereas rats that have not been deprived down here is 6 hours. That's very little deprivation in comparison. Their leveling off point is about 8 cc's. We call that a particular value, we named that as maximal intake because it appears that it's influenced by the amount of deprivation that you have. So it's an important number. And again, this switching point from the fast phase seems to be important. And the actual rate of quickness at initial quickness, that appears to be the same. All of the rats appear like they drink the water quickly at the same rate. It's not dependent on the different deprivations. It's just how long they continue that behavior seems to be affected. This is another image. Again, I think this was in rats that were given different types of food with different palatability. These type of studies have been replicated in humans, and what you see is that while the Emax doesn't look like it's different, it does look like the initial rate of increase is different for the different palatability. So what this means is that the rats that got some tastier foods ate quicker at the beginning than the rats who didn't. And I do want to point out, I'm going to bring this paper back up. This is a paper by Davis and Levine published in 1977. It is a mathematical model for the control of ingestion. It's exactly what we're trying to do today. And what mathematical modeling does is it takes all the information from experimental data and tries to encompass it within a formulaic viewpoint. And so it becomes kind of like a review paper, but even stronger than a review paper because it has to understand how all of these different experiments interact with each other and what they mean relative to a whole predictive value. And so I'm going to bring up this Davis and Levine paper again because not only was it a review of the literature at the time, but it was also a really forward-thinking type of model for the time, which is in, again, 1977. This was a paper on the number of licks made by rats during a 3-minute period. With some of the rats were injected with insulin and some were not. And you can see that over time, this was really a glucose concentration of what they were drinking, not versus time. But you can see that there's different kinds of curve shapes depending on whether insulin was injected or not injected. So this was, again, an older paper. I don't have the reference on here, but you can see that looking at this graph, that back then they didn't understand quite what was the impact of insulin on glucose concentrations that were consumed, and they were trying to understand it. Not too far back from these articles, we didn't really even understand what type 2 diabetes was, and individuals who had type 2 diabetes actually died from type 2 diabetes. So we didn't have any idea of how to control things. So this was the first kind of understanding we had of the interaction between insulin and diabetes. Humans, you can't lock them up in a cage and give them a lever with pellets given to them. That would be one heck of an IRB to prepare. However, we do want to understand some of the same things we looked at in animals for humans. And so a device called the Universal Eating Monitor was developed in 1982 by Harry Kiseloff. And what the device does, this is a modern day version of that device. There's a scale and a plate sits upon the scale, and the food is weighed. And as the person consumes the food on the plate, the grams on the scale are recorded and the cumulative intake curve is generated. This is actually Harry Kiseloff's paper. The Universal Eating Monitor was used for the first time in humans to understand the differences between ingesting solids versus liquids in humans. Now, what was interesting about this paper was I dug up this paper many years later after Corby first talked to me about the parabolas. I saw the parabolas for the first time in this paper. The reason I picked up this paper was because I was developing the position statement for the Obesity Society on consumption of sugar-sweetened beverages. And there's a prevalent belief in our society that we drink more calories if it's consumed as a liquid instead of a solid. And that's why I had dug up this paper, and that's when I understood what the parabolas were all about. And that's when I really started getting into this eating rate area. Harry Kiseloff was a pioneer in the field, and he is still at St. Luke's in Columbia University, where the New York Obesity Research Center is housed. So much of our understanding about obesity comes from that New York Obesity Research Center right across the river in Manhattan. So you can see why they would curve fit with a parabola. These graphs are not that much different than the graphs of the rats. Actually, if we go back and look, here's the graph of the rat, which looks very similar. to the graph of the human. And they look concave down, just like a concave down parabola. So the idea was, why don't I just curve fit this by a parabola? Why would someone want to do that? Why would someone want to curve fit with anything or do any modeling? Well, one of the reasons is that we would like to compare different cumulative intake curves with each other. You saw that the rat's cumulative intake curve was very similar looking to the human's cumulative intake curve, but it cannot be the same because we're eating solid foods and they're drinking sugar water. It can't possibly be the same curve. They differ by numbers we call parameters. For example, the Emax that I told you about earlier, the maximal intake. That number was different in the different water-deprived rats. And it looks like the initial eating rate might be different. We saw that in the palatability graphs of cumulative intake curves in the Davis and Levine paper. So we would like to have some key numbers that just tell us about the curve itself, so I can compare different individuals to each other or different populations to each other. Rats, humans, different kinds of humans, like maybe humans that have been affected by obesity versus lean humans. So there will be different things that I'd want to compare. We also saw that there was key timings of events. For example, in the rats that were deprived of water, that's a very important graph actually, we saw that the rats that were deprived of water for longer had a longer fast phase. So when that fast phase ends is a key timing point, an event, and I would like to know when that happens. So a model can tell us that, and I'm going to actually show you that. And obviously, I'd like to predict what happens beyond the observed data. Someone may sit and stop. Those of you who have kids know that when they say they're very hungry and they sit down to eat, they really go through the rapid phase and they want to get up and leave and play. You would like to say, look, sit down and keep eating. Well, what would happen in humans and adults when they stop eating? What would happen if they just kept eating? A model should be predictive. Most models are predictive. That's like the 101 of a model. So they want to predict beyond the observed data and say what happens. So I want to introduce you to first principles modeling, and I actually want to give you a pitch for something called first principles modeling against curve fitting. It's not that I'm against curve fitting. Curve fitting has a time and a place. This is not the time and the place. So what is a good model? What is the definition? What's amazing here is that in the literature on cumulative intake curves in 1977, McCleary published 4 criterion which define good modeling, which is amazing because in modeling literature, as a modeler, I have learned those 4 criterion, and there's a book by Batzel, they're mathematicians, and they actually have listed out those same 4 criterion in a more general setting. So this is a standard 101 for modelers, but that it appeared in early cumulative intake curve modeling papers was pretty amazing. So what are those criterion? First is that the model should generate realistic predictions beyond the observed data. That's the first criterion that we have. That's usually what models are used for. Maybe we want to predict obesity prevalence, or maybe we want to predict the weather 5 days from now. The models are what give that to us, and we would like to take the observed data and predict past that observed data. The second criterion, which is sometimes misunderstood, is model simplicity. You shouldn't make the model include every possible reality because then it becomes what we call intractable. We can't get anything from it. So there should be a balance between including enough of the biology, but not including so much that the model becomes overly complex. That doesn't say that it should be overly simple and invalidate criterion 1, for example. So there should be a balance between all these criterion. The model itself should provide us with some mechanistic insight. We should learn something from the model that we didn't have from the data itself. The final criterion is really difficult, and that's that the model should be based on complete theory and that there should be no nonsensical predictions or no parameters that give you nonsensical predictions. Everything should make sense. Models that satisfy this 4th criterion mean they give you the curve as an outcome. The general shape of the curve as an outcome, not a priori knowing the shape and then fitting it to curve fitting. That's where curve fitting and model criterion number 4 differ. McCleary in his 1977 paper said, to that date, they had not achieved criterion 4. What we're going to do is we're going to develop a model that also satisfies criterion 4. We're not the first to do this. 1977 paper by Davis and Levine were much more advanced for its time, and they had used circumstantial evidence at the time to say that there must be something that starts gustatory stimulation, that initiates it, and that the feedback from the gastrointestinal tract must control that ingestion and give it some negative feedback so that we stop eating. And so they were using the data at the time to make these conclusions and develop a first principles model, which is pretty amazing. And I just wanted to show you, because you see the history of what we were learning. We know today that gut hormones like CCK, which is one of your learning objectives, these gut hormones are the regulatory mechanisms that instigate gustatory stimulation and inhibit as a protective mechanism, as they say here, must be the case. CCK is what inhibits, ghrelin is what stimulates. So we know these things now, but they didn't know it then, and they were guessing and arriving at that conclusion through circumstantial evidence. So you see the history of understanding the gut here in a modeling paper. Unfortunately, in 1982, Kiselov, who again I say was a pioneer in the field, but he also was an inhibitor of the field as well, because he advocated for the use of just curve fitting. And curve fitting it has been adopted by the field of eating rates and is used today prevalently. And it was because of these initial papers by Kiselov which promoted the idea of using quadratics to model the cumulative intake curves. So the 1977 paper of Davis and Levine never took hold and never was progressed past that. And you might ask, why? Well, one of the things in medicine that people like is they like rules of thumb. There's many rules of thumb in nutrition, and individuals that advocate for the rules of thumb say that it should be easy. But again, we are looking at those 4 criterion, and they should be satisfied. And the model should not be overly simple so it defies those other criterion. And that's the problem in using a curve fitting approach only and not allowing modeling to advance. Here you see that the reasoning Kiselov has for not using the Davis and Levine model is that it's nonlinear and difficult to apply. This might have been true in 1977, but not true today, where we can build apps, and we do build apps so that people can we can deliver these predictions clinically. This is really, really important. There's many complex models that are out there. For example, the models that model the weather and the atmosphere are really, really difficult. They're highly intractable. However, we use those predictions on a daily basis to even decide what clothes we want to wear. And they've found a way to deliver those model predictions with sunny faces, cloudy clouds and rain picked images so that we can decide what to wear. So we never have to look at the nasty equations. We just look at the delivered information. We have to think in medicine a little bit along these lines because being overly simple and giving incorrect prescriptions and incorrect and inaccurate advice is not good, especially when it comes to medicine. So why would a quadratic not be a good idea to use? I'm going to give you some examples and reasons for that. First, this C in a parabola, if you don't remember parabolas, that's okay. I'm going to show you this pictorially in a second. But this C represents the initial amount of food consumed at time equals 0. Theoretically, that should be 0. This is what we call a fudge factor. So I explained what a fudge factor was scientifically to my little girls who are 9 and 10 years old. And then I asked them to relay back to me what a fudge factor is. And my 9-year-old said, a fudge factor is something that you have there just to make things look good. And that's true. It has no biological meaning. It's just there to make things look good. So theoretically, that should be 0. However, it's not set to 0. The best fit is not set to 0 because it has to look good. B, the second parameter, is the initial rate of food consumption. And this parameter does have meaning. After you do the curve fit, you can compare the B value of B for different cases. As we saw in the palatability of the rats, the B should be different for the different types of foods that were presented to the rats. And we know from Kisselhoff's work that the B is different for different palatability of foods presented to humans as well. Another assumption here from the parabola is that the acceleration of food consumption is the same across the board. We know because there's a fast phase and a slow phase that that's incorrect. So there's some limitations to curve fitting by a parabola. You will never get that inflection point that we see with a more sophisticated model. The fudge factor, Kisselhoff himself says it's simply a curve-fitting artifact. For all intents and purposes, it should be 0. The best fit, though, is not 0. I actually took some of Corby-Martin's data, and I fit the best possible quadratic. That's the blue curve to the data. And in this case, at time equals 0, it gives me that there's a negative amount in the gut, which makes no sense at all. Now, you could force that intercept to be 0. You get a less good fit, which is the red curve. And you either have to live with that not as good fit, or you have to say, I'm going to use the fudge factor and force a good fit. So that's a problem. I want to show you another problem, and that's what we call nonsensical predictions. This came out of an image from a paper by Gust and Kisselhoff, and you can see in this image, he's fit a parabola forcing the 0 to intercept to be 0. But here at the top, you see this dash curve. The parabola eventually cycles downward. And so here we get nonsensical predictions after 5 minutes because cumulative intake should never decline. You're always consuming more food in your eating duration. And so the cumulative, it should always be an increasing function. But parabolas are not increasing forever. And so eventually you will get nonsensical predictions. So that also is a not good result of using a parabola. So if we look at those 4 criterion, the parabolas do not generate realistic predictions beyond the observed data. We saw that from the image you just looked at. It is a simple model. However, it was simple at the expense of criterion 1 and some of the other criterion. It does provide us mechanistic insight. For example, I can take that value B, estimate it for different individuals, and see if, for example, someone with an eating disorder has a different value of B than someone without the eating disorder. So I can make comparisons. So it gives us some mechanistic insight. But it doesn't give us full mechanistic insight because we don't have the inflection point, which we definitely see. Is it wholly self-consistent and give us the curve as an outcome of the model? No, it completely fails this. Remember, the generated observed curve shape should be an outcome of the design model, not curve fitting. And so it completely fails criterion 4. In the model that we develop, we begin with a theoretical basis. So we're looking at criterion 4, and we say, look, are there 3 assumptions that we can come up with, and are there justifications for the assumptions, and then build the model on top of those assumptions. The first assumption is that eating rate is dependent on the amount of food eaten so far. Well, we not only know this from looking at the curves themselves, that so there must be some signal, but there is this beautiful studies, which unfortunately ended up with the mice dying, but what we know is from sham feeding studies. That means that mice were fed glucose water, but the glucose water was drained from a opening in the throat, and so that although they felt like they were drinking the water, they were not drinking the water. It was not reaching their gut. What we know from those studies is that the mice kept eating, and so there is some kind of gut signal that influences the rate of eating. And so there's evidence for this. We also know that in the initial stimulation of eating, that the eating cumulative intake curves are linear. We can see that from the curves over and over again. It's in every single cumulative intake curve. So those come from cumulative intake curve data, just looking at them. And the third is that there's a second phase of eating after this initial phase where satiation kicks in and the eating rate slows down. We know today what causes that. That's CCK, for example, which is a gut hormone. Kiselov actually had pointed out that food intake is self-limiting, which is not reflected in the parabolas. However, we would like to have that as a theoretical basis. We know that from the CCK injections, Kiselov actually ran that study, that infusion of CCK resulted in preservation of the cumulative intake curve, but reduction of what we call Emax, the total maximal intake. So here's what our model looks like. It's not a curve fit. It's what we call a differential equation. The eating rate is represented by a derivative. It's the rate of intake as a function of time. Theta represents the initial eating rate. It's the same as B, the value of B inside Kiselov's parabola model. The R times E represents that linear phase that you see at the initial phase of intake. So the dominant term is that first term in the product, which represents that initial linear phase. However, as E of t, which is the amount of food consumed at t minutes, reaches an E max, the inhibitory phase begins right here. So that second term starts to dominate. So as E of t gets close to E max, this fraction gets close to 1, and this term gets close to 0, and the eating rate reduces to 0, and it flattens. So we get the self-limiting nature of eating, the cumulative intake curves built into this model. So everything is there. The R is hard to describe. You can do some calculations, which we do in the manuscript, which is now under review at the American Journal of Clinical Nutrition. But 1 over R can be thought of as the time it takes to effectively double the amount of food consumed. Just to show you pictorially what these numbers do. So if I took data and used the model that I just showed you to fit that data, then I can estimate what the Emax are and theta are. If I increase Emax from 400 to 600 grams, and this is data that actually came from Corby Martin's study, which I'm going to show you in a second, but you see that increasing Emax increases that self-limiting value. This looks very much like the water-deprived rats, where Emax was increased. So I can actually see that reflected in the curve. Changing R, or increasing R, what that does is it changes the mid-level of the curve. Right here, you see that the mid portion of the curve starts increasing. We did see that in the palatability graphs, and we even see even more in the palatability graphs that theta, which is the initial rate of eating, increase. If I increase that, I see something that changes like the palatability graphs that I saw with the rats. And so these three parameters, Emax, R, and theta, completely describe this curve, and they all have biological meaning. None of them are fudge factors, and we have no nonsensical predictions here. Everything, you'll never have declines. If someone continued eating as in a pizza eating contest, you will get this leveling off. It reflects your child's eating when they eat really fast at the beginning and they start to slow down at the end. So you really get everything you can and every shape you can that is already reflected in any one of these cumulative intake curves. So what's good about that? And so what? I have a better model that satisfies the criterion. What can we do from that? Well, we can ask some questions. The first question we can ask is about the hypothesis behind the book cover that I just showed you, the very first slide I showed you, where mindful eating is supposed to contribute to less eating. So behind that hypothesis is that individuals with a higher body mass index may eat faster and are not mindful. That's kind of the presumption behind that hypothesis. Well, we can test that. We can test that using data. Does body mass index actually influence the cumulative intake curve? If it does, I should see that cross-sectionally, that body mass index is a influence on the shape of the curve. And it should affect those parameters. So I can write a model actually that predicts the theta, the R, and the Emax from gender, body mass index, and age. The second thing is, if I took someone and changed their eating, can I just change their intervention, the parameters, and then dictate to them how they should eat? That means, does the parameters themselves encompass entirely that curve when I alter that curve in an individual? And the third thing is, can I use those model parameters to distinguish between different eaters? For example, I had mentioned earlier that Kiselov had shown that the parameters in the models differed in individuals who had eating disorders. So could I do the same here with these parameters? Do they tell us something different when someone's a restrained eater versus unrestrained eater? And finally, the most important thing that we have here is that we actually can tell you when the eating switches from fast phase to slow phase. So when does that happen? For this, I need data because I need to look at people, actual people, and calculate their theta, R, and Emax, and I need to look at those relationships. And so we're back to Corby Martin's original study on where he was talking to me about the parabolas. And what that study was about, it's actually a fascinating study. They took 48 subjects. It was in a randomized controlled trial, and those subjects were first asked to eat ad libitum, so they had a baseline meal, and their eating rates and cumulative eating curves were recorded using universal eating monitors. Then they slowed down their eating by 50% by using a bell to tell them when to take the next bite. So what they were testing for in the study was whether slower eating was going to reduce the amount of food consumed. Slightly reduce the amount of food intake in men, but did not in women. So these gains were not that much for the amount of work it was. And that's by verbal. That's one of the only times I've seen Corby Martin irritated. He hides his irritation usually, but he was irritated because this was a lot of hard work for very little gain. So that was the punchline of that story, but I can use his data to answer my questions about the modeling. So we did this. We tried to predict initial eating rates, the value for R, and the value for Emax by using what we know about individual characteristics. And we hypothesized initially that body mass index would predict these parameters. Unfortunately, body mass index was not a significant predictor for any of the parameters. So there goes the hypothesis behind mindful eating. Body mass index does not look like it contributes to the cumulative intake curve shape. Well, that means that there's characteristics that dictate the form of that curve, but we don't know yet what they are. We can tell you something. For example, males and longer meal duration were associated with higher values of initial eating rate. Second, females were associated with higher values of R. That means it took them longer to double the amount of food consumed than males. Males were also associated with higher values of Emax, what they maximally can consume. And in this Emax, BMI was borderline significant. The p-value was 0.08. So it shows that it might be influential. I don't have the data for this yet, but I quickly checked whether body mass index was a predictor of the timing between fast to slow, and it is. So the end conclusion is we need to dig more. It's not just something very simple that explains the shape and the form of the cumulative intake curve. Still something out there to understand. Are these values related to each other? So for example, if I eat really fast at the beginning, does it affect how much maximally I can consume? It does. So you see Pearson correlation constants here. Emax at baseline is related to theta at baseline. That means if I eat really fast at the beginning of my meal, I maximally am going to eat more. That is interesting. So there's some other interesting facts that we have here. What about once I reduce the amount of food that's consumed? So these are the reduced rate thetas, R's, and Emaxes, and are they related to the baseline? And they are. So remember, in Corby's study, he took individuals and reduced their rate of eating, and he reduced it by 50%. And you see that the baseline and the reduced rate were related to each other by a fairly high Pearson correlation constant. So what we're seeing is that, yes, indeed, I could just manipulate these parameters. If instead of using a bell and ringing the bell, I could present the individuals with their revised cumulative intake curve after revising their parameters and use the revised curve to guide their eating if I want to do that. Although the results of Corby's studies suggest that we may not want to do that. There's another study by Westerterp-Lagenta who looked at unrestrained versus restrained eaters. And what we did is we calculated the Emax and R and theta for each of the two groups. The two groups were directed to eat as much as they can and as much as they feel comfortable with. And what you see is when they're asked to eat as much as they can, their Emax goes up, as it probably should, right? Because they're tasked to eat as much as they can. In general, the Emax for the restrained eaters were higher than the Emax for the unrestrained eaters. And the Emax in the eat as much as you feel comfortable with was much higher than the Emax when they were directed to eat as much as you can. So that answers the question of whether I can distinguish between types of eaters and types of directives using these parameters, and the answer is yes. Notice here that the theta, which is the initial eating rate, is fairly stable in all of these. What does this tell us? Well, there's a longer fast phase in the unrestrained eaters, and once they're directed to eat as much as they can, they have a higher max capacity of eating. Like the deprived water, rats that were deprived of water. What about modeling the switch between fast and slow when that happens? I can actually calculate it. I couldn't calculate it using the parabola because the parabola has a constant acceleration. So that means there is no switch between fast and slow. But I actually can calculate it here. And what you notice, this is one of your learning objectives, what you notice is that the time switching from fast to slow is a function of your initial eating rate, your doubling time for food consumption, and your maximal intake. All of these parameters influence when you switch from fast to slow. I can actually graph this because it's hard to see all those parameters in one formula like that. So graphically what you can see is if I hold theta fixed, which you saw from the restrained and unrestrained eaters, theta was fairly fixed and fairly constant. So I held it constant at 50. What I can see here is that the longer it takes to switch from fast to slow, the higher the Emax. That means the maximal intake. And also we see that the lower the R. So the lower R means that the doubling time to eat the food consumed has gotten higher. So again, all of this makes complete sense, but now I have a formula that relates them all together. And it's not a linear, this is not a linear relationship. As you can see, this relationship is curved, it's a curve. What is very interesting to us is that there were regions, parameter regions of R and Emax where there was no inflection point. So what that means is here with low Emax and low R, there was no fast phase. There was only a slow phase. And I like to believe my daughter has this. None of Corby Martin's study participants had this. So I think normal eating behaviors in this range, maybe this range here has nobody in it. everybody appears like they have a fast and slow phase. So over here in this parameter region, there's both a fast phase and a slow phase. So I just dug around and checked. I also dug around and checked whether body mass index was a predictor of inflection point, and it is. So the time it takes to switch from fast to slow, BMI was a significant predictor, weakly significant. It was a weak predictor, but it was a predictor. So there we have our first inclination that body mass index might influence something in the cumulative intake curve. So these are some of the conclusions we can make. I want to thank you. Listening to a math talk is not easy. I want to also thank my Montclair State University team. We have a great team of researchers at the institution itself. My collaborators around the globe, especially Corby Martin, who had initiated this conversation years ago and stayed friendly with me so that we could continue this on, and my NIH funding, which allows me to reduce my teaching load and work on these very interesting topics. Questions?